This is a collection of slides from me, Sergio, and Marco from our group. And stop me at any point to ask any questions. If anything is unclear, I would love to elaborate on as far as I can, at least. So the basic kind of process that we follow here in this lab can be kind of broken down into specific repositories that you can find at our GitHub. So it's basically a collection of different repositories dedicated to different things. So first of all, I hope you have had a tiny introduction to ISS, or if not, I hope you can follow either way. But basically, so we have a dedicated kind of repository to each of these steps. So, but I will mostly focus on the image processing and pre-processing of this. So there will be some image acquisition, of course, happening. So as you see now with this base text format, this images come from somewhere, right? So we have a biological tissue that we want to take a look at the spatial gene expression of some predefined markers. Then we acquire these images. And there's a few different things that we do. And now you got a very brief touch on this kind of deconvolution-based approaches to clean up these images that have been acquired. So that is sort of a part of our pre-processing workflow, but they are not mandatory, right? I mean, you can still manage without them, but they do generate better data in general, as far as we've seen. So, and then there's also something called CARE, which is also a type of image kind of reconstruction or deconvolution, but it's not an actual deconvolution in the sense that a flow deck might be, or something like this based on a PSF. Rather, it's actually just a neural network that learns based on previously deconvolved data. So that is basically it. So I'll run through some of these steps just to give you a feel for what you can do with the data. And again, everything is on the MolDIA GitHub. MolDIA stands for Molecular Diagnostics, which is not really what we do these days anymore, but this has sort of been a relic of what we used to do in the lab, which was more diagnostics. Now we've more moved into kind of this spatial sphere. But in any case, you find everything here and can, of course, post issues and anything and contact us. It's not the problem. So first of all, I would just briefly mention deconvolution and restoration and what kind of that entails and what you can expect from it and why we would want to do it. So the whole point of this is when you do look at your images that you have acquired from the microscope, there is some inherent blur in the images that you have acquired, and that is because of those kind of optics that's in there. So a point source or a spot won't be exactly a point, rather it would more it would be more of a smear or spread. And this is what's called a point spread function. So the whole idea of doing a deconvolution is to kind of leverage this information that you have about your microscope setup and try to restore or kind of improve on your images. So the aim of this step is to try to improve the quality of your images and produce better signal-to-noise ratios. And this will, in the end, give you more data to work with. You get more decoded spots, you get higher quality measures and metrics, and this will also improve the decoding performance on the low magnification of your dense data. This is at least what we've seen in our lab, and it does indeed work very well. So the kind of routine here is that you need to define your point spread function. So you need to have the information about the microscope that you acquired your images on. And then you apply your deconvolution method of choice. And in our case, we use something called flow deck, which might not be the best, but it works quite fine. There's other ones called Deconvolve, for instance. And many of them work on GPU acceleration, which sort of makes the process a bit faster. So basically what you do is that you move from something on the left here to something more on the right, where everything is more kind of well-defined in your images. And then I told you about this CARE, which is something a bit differently. It's very interesting in a sense, and to me it's more or less just black magic, because you kind of train a model based on your images. So you have a training set of images, and then the machine can kind of replicate what it sees from your model onto your new data. So the aim here is actually just the same as a regular deconvolution, but the whole idea is that we do it in a fraction of the time. It offers similar benefits to deconvolution, which is then more spots and better spots, but it's of course not as accurate. It's of course an approximation, but it's still pretty good. And you have to really make sure that what you are indeed kind of clearing up or making better is not just an artifact. I mean, if there's a spot there from the beginning, it should be a spot afterwards, and it shouldn't invent kind of new spots. And it's roughly 100 times faster than FlowDeck or any type of deconvolution that we have running in the lab. And this is kind of why we want to do this, right? Because a proper deconvolution can take time. And the routine here again is that it requires a trained model. We do have one from the lab that you can download and use. Of course, we claim no responsibility at all. So please always double check everything to see that it's okay. It uses the GPU, otherwise it will be very slow. And again, the idea is to move from a blurry projected image to a sharper projected image. So basically, the training set that we have in the lab currently is 3 groups of images. It's raw data, it's spin data, and then deconvolved data. Images from both of the microscopes that we have in the lab, Leica and Zeiss, and 20 and 40x, and then all channels, not API. So again, we claim no responsibility here. We just think it's a great tool to use for people in the whole image-based spatial transcriptomics sphere. We have multiple tissues and organisms. It's flies, mouse, and humans. And we included some binned images just to improve the accuracy in very noisy data. So now on to the actual kind of pre-processing of our data. So when I say pre-processing, I mean that we move from the raw images that are required on the microscope to something that we can at a later time point put into something like starfish or something like this. And you'll understand why in a minute, why we need to do this pre-processing. In an ideal world, we could just use the raw data from the microscope and get our spots decoded in space. But of course, life is not that easy. We usually have to go through some sort of steps to make this possible. So the aim here is, as I said, to go from raw images from microscopes to something that we can decode and identify each spot. And this is achieved by aligning the sequencing cycle, as you just heard from Christophe. And we also need to remove, at least in our case, we need to remove this 10% overlap that we have between imaging fields of view, because this is what we generally do. We acquire images in this field of view type of fashion where you don't, it's not possible to image the whole section in just one image, basically. Rather, you have to break it down to smaller images. So that's what you need to do. You need to remove this overlap, and this is achieved by stitching the image and aligning it. And then we retile it to kind of reduce the workload of the computer, and you'll see why. And the routines here, they're present in a notebook for examples, so you can have a look at that and try to see if it works. But in this course, you won't really, I think, do like go from raw images from a microscope to something that you can decode. And the technical aspects here is that it's using something called Ashlar, which was published quite recently for cycle alignment and image stitching, which is great. It works wonders, really. So again, what I try to get across now is that each single field of view like this, let me turn on my laser pointer. So each field of view like this needs to be stitched or sort of put together in a fashion that allows us to get a full image of our entire tissue and each cycle and each channel. So the way that to kind of break down our data is we have roughly 5 cycles of imaging. It's kind of a usual case. Sometimes you can have more and sometimes you can have less. We usually have 5 channels. So one channel would be for the nuclear staining and the other ones would be for fluorescent dye to be able to decode the identity of our channels. Of our spots. So 5 cycles, 5 channels, and then any sort of set stacks that you have. So we usually run from anywhere from 11 to 21 or even more. That amounts to a lot of images, right? So the bigger your section, this kind of increases exponentially. So you get a lot of data. So that is kind of the main problem that we have in the lab currently is sort of how do you store all of this raw data? You can't So the way that it's stored now is just on a ton of hard drives. Like the best scenario would, of course, to have some sort of server somewhere to store everything on, but currently this is where we're at. And again, as you've heard today previously also, is that images are basically just matrices, and values in these matrices are represented as intensities. So the pre-processing, again, works by firstly taking all of the Z stacks that I mentioned, like the 21 Z stacks, and for us, just merging them into one. So basically, for each single pixel, we look at the most intense pixels across this 21 Z stacks. So we want to project that into one. Then we extract some metadata to know and have a feeling for where these fields of view are located in space. And then we align and stitch using ASLR. And then we reslice the stitched images, and that is the kind of starting point for our data. So we can do then to use Starfish for this, the SpaceX format, and then Starfish to decode. So yeah, I'll just briefly go through this. You'll probably have some more input on this later, but I just wanted to give you kind of the whole workflow. So in the ISS decoding stage, what you really want to do here is you want to detect each single spot and then decode this identity, right? So look through the channels and sequencing cycles to make to kind of decode the identity of this spot. So this is achieved by formatting the data into something SpaceTX compatible, which is what we talked about previously today. We do some top hat filtering, we normalize the images, and we detect spots from all cycles and all channels. And then we record basically the intensity of each of the single spots in each channel and then cycle. And the routines here again are the stuff that you can find in the GitHub repo, and all the technical aspects you can read about in Starfish. But it's I believe, or I think it's a great tool because it offers a lot of functionality in this sort of same format. So you don't have to kind of format your data to be able to be compatible with other softwares. Rather here, it's all collected into one main thing, which is great. So yeah, the decoding works as follows. We take the SpaceTX format, the data, we can create a reference image by, in essence, projecting all signal images into one. So you create this sort of anchor image of all your spots. So this allows you to define the spots, and then you can decode the intensity. And then you can do some extra alignment that Christophe just talked about. We, of course, do the alignment prior to the steps. And then you can also go by normalizing channel intensities and localize the spots, and then decode the spots. So now I just want to show you a very quick example of what you can do with ISS data that is very close and dear to me. This is sort of my main project. And it's a mouse model of EAE, or sort of multiple sclerosis, called the EAE model. And as some of you may know, multiple sclerosis, or MS, is an inflammatory demyelinating disease, and it's a horrible disease that really causes issues for people. And the EAE mouse model is sort of trying to induce these characteristics in a mouse. And the prime kind of characteristics of MS is inflammation, so this enormous infiltration of immune cells into specific regions, and then the breakdown of the myelin that sort of surrounds the axons, and this causes axonal loss, and it's a really horrible disease. But so the idea here for us was that we wanted to kind of look at the effects of this disease, or the model of disease. And we wanted to look at the disease stage, CNS region, and possibly the sex on kind of the distribution of a few select cell populations. We wanted to look at these areas where the immune cells are infiltrating, called lesions, and then look at the lesion composition. We also wanted to look at primary cell-cell interaction based on proximity, which is great, right? This is why we kind of do spatial transcriptomics, is to be able to actually see where all the cells are. And we also wanted to look at disease progression and sort of at the peak disease and at the later stage to see what's happening in the system. And then the idea for us is to kind of classify the lesions based on differences in the cellular composition and the location where they're in the CNS, and then the disease stage and sex, and correlate it maybe to human lesions. So all in all, the project can sort of be boiled down to this. We had a total of 239 genes that we selected for to mark basically a few primary cell populations. So it's oligodendrocytes, or this kind of very do-it-all things in the nervous system that they sort of facilitate the functions of the brain and neurons. So we wanted to both look at disease-specific oligodendrocytes or lineage cells, and then also these general types of cells. We also wanted to look at immune cell populations, right? Because this is a inflammatory kind of disease where these immune cells are really infiltrating the areas of the spinal cord and breaking down the myelin. So there are, of course, immune cells in the brain and the spinal cord as is, but we wanted to look at both the non-resident and the resident cell types here. We also wanted to look at astrocytes, immune astrocytes and regular astrocytes, neurons, other vascular cells, and Schwann cells in the immune. So this is kind of the outset of the whole project. And the data set collection has thus far has been 18 total spinal cords and 8 brain hemispheres. So they're sort of broken down into these different regions of the CNS. So we have lumbar, thoracic, and cervical, and then the brain. And then we wanted to look at kind of how the disease is affected across the CNS. So all in all, it's quite a big sample. I think we're closing in on 1 million cells total. So it's quite an extensive data set. That it's great that there's tools out there such as ScanPy that we can use to actually analyze these cells, because that is one of the main upsides with using sort of spatial transcriptomics. It's not only that we can treat it as single cells as we can by segmenting cells and then assigning spots to them, but also we can also leverage the spatial kind of localization of each of the single cells. So this is kind of what we're left with. So this is a lumbar region of a EAE-induced mouse. So this is the gene expression. You can see kind of this, hopefully it's semi-high res on your screen as well, but you can see this kind of very densely packed regions of the spinal cord here, which is where you get a lot of expression of immune-specific cell markers. So this would be kind of a, so where you have this increased density, this is probably where you have the lesions, right, where these immune cells are infiltrating. Then you see other types of cells here in the gray and white matter that we also map out, of course. And then we can actually use programs such as ScanPy or things like this to actually do some dimensionality reduction, in this case a UMAP, and then we can cluster later on. So in this case we have, and as you can see, it's not as sort of clean as a single cell data set would be, right? So there's some more sort of mixing happening here, but to some extent it's very clean. You have this main type of populations where you have neurons, you have oligodendrocytes, you have astrocytes, these Schwann cells, you have some early oligodendrocytes, you have other types of neurons, these vascular cells, and then the immune cells, and then the other T cells are located down here. So now the point is to kind of grab this annotation that we've done and then map it back onto space, right? Because that is what we're, in essence, very interested in, because we want to see what happens over time. So this is one example. So this is looking at what we define here as the primary neurons, or the first neurons, and then you can see, so just to kind of decode this for you. So R stands for round. So round 1 would be sort of the first collection of experiments that we have. And then 2 would be then correspond to 2, etc., etc. Then C would stand for cervical, which is the region of the spinal cord. Lumbar is L, thoracic is T, and then you have EE or control. So we had coupled disease and control. So you can kind of map this out this way to sort of get a feeling for the immense data set that we have here, right? So there's a lot of cells, and then we also have the spatial organization stuff. So you can see that the neurons are very well sort of conserved throughout all of the tissues that we've collected. Now I'm not showing any brains, but this is just one, right, to give you a feel for the data. And then we can start to look at these Schwann cells and the meninges, and you see it's also very well conserved across the tissue sections that we have. You can look at the T cell population, and you see there is this kind of enrichment in the EE sample. So you see this EE compared to the control, it's a great increase in the amount of T cells populations here. This is even more if you just look at the immune cluster in general. You see very clearly like what sections are in the immune, or sorry, are in the disease and what are in the control. So it's a very nice way to look at it. And then we of course also have the exact spatial location of each of these single cells, that we can then start to look at neighborhoods of each cell to make sense of how this lesion are sort of comprised and what biological processes are happening. And then, yeah, that's more or less it. I'm open to any questions, and this is, yeah, just to show you basically a very interesting project that we're working on using spatial transcriptomics.